Okay, guys. Thank you for everybody for being here today on Zoom. I have the pleasure of introducing Dr. Christopher Malinowski, who comes to us from Ocean's First Institute, but he started his education at University of Wisconsin, Stevens Point, and then upon graduation, he worked in Fiji colleges up in Wisconsin, and then finally found his way down here to South Florida, doing a master's at FAU, and then getting his PhD working with Goliath Grouper over at FSU. And now I would describe Chris as just the quintessential coastal ecologist. He's done everything from deep ocean work to work terrestrially, as well as just along the coast in general, and everything from pharmaceuticals and toxicology up through population dynamics of these big megafauna species, such as Atlantic Goliath Grouper, sea turtles, and dolphins. Now, he comes to us from Ocean's First Institute, where he is now a nonprofit, where he is the head of research in a newly created branch down based out of Key Largo. He's been there for about 2 years, and he is also an affiliate research professor up at FAU, and he is becoming an affiliate research professor here at FIU. He's doing that right now. So please, everybody, welcome Dr. Christopher Malinowski, who's going to talk about Atlantic Goliath Grouper, which are some awesome fish. So
Thank you, John. It helps to have a friend with your introduction because you did that better than I would have. And also that means that I can cut down on some of my introduction about myself too. I actually had a whole plan to talk about more of my research today, but I had so many slides in here about Goliath Grouper. I decided that was going to be the primary focus. So if you will, it's actually a pretty cool topic to talk about. I think it'll be entertaining as much as informative. So I just want to give a little bit of an overview of some of the things that are important to me. And I think I'm a member of the IUCN Species Survival Committee for groupers and wrasses. And I think what they embody is kind of what we all are sort of after, which is, here's a quote from their website, which is to seek a just world that values and conserves nature through positive action to reduce the loss of diversity of life on Earth. And I think that's something as ecologists, scientists, as humans that we should all strive to try to accomplish. And so some of the responsibilities for that position and just overall as a responsible ecologist and scientist are to do things like, evaluate statuses of especially exploited species, develop action and management plans, strive towards equitable international resource use and fair trade, evaluate data deficiencies, which occurs throughout the oceans, right? And then also education becomes a very important part, right? We can do all the science in the world, but unless you can communicate that science to the public or to the audiences that need to hear for management or whatever the case may be, then it can get kind of lost, right? So those are all sort of important things and sort of embodies the reason why I have become an ecologist and a scientist. And that sort of then transitions into my current position, which is Director of Research and Conservation in this small nonprofit that John mentioned, Ocean First Institute. And our whole model is to work under the umbrella of conservation through research and education. We sort of hit a bunch of different pillars of that and allows me to do a lot of the research that I care about. And so throughout the course of my career, I've done a lot of different things, right? From undergraduate degree all the way up through where I am now. And that has taken me into a bunch of different environments, from offshore around the world to working with big Goliath Grouper that I'll talk about primarily today, to the Everglades, working in estuaries around Florida and species like snook. Started off in marine mammals. When I left Wisconsin, I thought, the farthest thing to working in the Great Lakes would be working with marine mammals in the Bahamas. So that's what I did for my master's research. Of course, then I eventually found my way back to the Great Lakes during my postdoctoral research, but that's a long story too. So a lot of cool things, and I think the story is that there's just a lot of interesting things to study out there, right? And for graduate students that are in here, you don't necessarily have to choose just one path, but you can sort of work within the realm of something you're interested in and go off in a bunch of different directions. That's what I would encourage you to do. So I'm going to talk about groupers today, right? Which means that anything in the family Epinephrilidae, right? And so far there's been described about 163 species, and that's worldwide. In the Western Atlantic, which is primarily what I'm talking about today, there's 22 that are described. And of course, that's constantly changing through new genetic information that we get from fish and differentiate them based on their genetics. And so, what's up? Oh, yeah, thanks. Good. If we dim the lights a little bit more too.
I think it's on the panel thing.
Oh, probably. Sorry. What are the relevant things? So groupers in general are sort of the fish that you think of when you think about like a robust, deep-bodied fish, big lips, reef associated benthic animals. They have a lot of really cool, unique characteristics that I'll get into some of today, not just morphologically, but also physiologically, right? I mean, just in general, they have spiny dorsal fins and fin rays that are soft behind those. They have really interesting dentition, which is why I put a picture up here too. They have sort of teeth that surround the jaws, and then they also in the bottom picture they have the pharyngeal teeth too. So these don't work to chomp and shred prey like a shark or something. But these actually trap fish, right? Or their prey. So they're engulfing prey. And then they're using those teeth to sort of trap their prey in there. So the pharyngeal teeth that are back in their throat actually kind of work as a conveyor belt to keep prey back. So think about like crabs or fish, they would gulp in, it wouldn't be able to escape. These teeth are all pointed backwards. And they also have quite a few characteristics that make them very vulnerable to overfishing or overexploitation. So I've listed a few of those here. So slow reproduction or production rates. So they have very slow growth, right, in late maturity. So for example, Goliath Grouper don't mature until about 6 or 7 years. So that's quite a long time until you can start adding back into the population. Warsaw Grouper, which is another very big deep water grouper, they don't mature until about 9 years, right? And then Gag Grouper, which is a very important fishery, especially in the northern Gulf of Mexico, not until about 5 years. So you can imagine that has a lot to do with when they're being fished out rapidly before they have a chance to mature and add back to the population. They're also generally very long-lived. Many form spawning aggregations that are annual, and it becomes very predictable. So for fishermen that know these sites, know the predictable aggregations that they form, they become very easy targets to go and collect as many fish as possible, especially if there's not as many restrictions in countries that don't offer a lot of enforcement or regulation, right? It's happened, you've seen with Nassau Grouper, Gag Grouper, there's a lot of groupers that have really have been overfished quite a bit, and especially their spawning aggregations have been fished out. They also, a lot of them exhibit high site fidelity, meaning they don't leave their sites very much. They're residential, so they don't leave those sites, and so for like a spear fisherman, you can easily target that. They use estuaries as nursery habitat, and so this is important because estuaries are extremely important for a lot of species, and they're also very heavily impacted, right? A lot of you probably work in mangroves or seagrass habitats. And we know that those are heavily polluted or developed, and Florida has definitely done a lot of this, right? Up and down the East Coast. I'd say the southern part of Florida is still pretty good in terms of the Everglades and 10,000 islands, but we really messed up a lot of our coastal habitats. And so those are heavily impacted by water flow and nutrients too. And so, for example, some of the fish that use those habitats are Gag up to 6 months in seagrass, and Goliath Grouper up to 5 to 6 years for early life history. This one is more specific to Goliath Grouper, but a lot of grouper are not afraid of divers or being approached. They're easy to approach, especially if you're a spear fisher. And then there's also negative interactions that'll happen where they'll start to steal bait or something dangling on the end of a fishing line or a spear fisher line, and that adds to a lot of negative interactions that people complain about. And again, that's very specific to Goliath Grouper. And then also they have a high value, right? So high economic value, high market demand. So when you go to a restaurant, you often see grouper on the menu. You don't know what you're eating, right? And most people don't ask what kind of grouper, what size. You just know that they are a high value, high market demand, and not knowing what that is from a consumer standpoint can actually be pretty detrimental. I'll also talk a little bit about this today, but sex change adds another dimension into this, right? Most, if not all, grouper are sex changers, hermaphrodites, and they can change as protogynous hermaphrodite is the term. They change from female to male, or at least have the potential to at some point during their life. And so that you can imagine how that adds into like a stock assessment if you are just looking at females, but adding the complexity in there of the sex change can really add some issues to that. And so looking at worldwide fisheries, this is a bit of an old statistic at this point, but it still holds from Sadovia and Mitchison, or Dave Mitchison et al. In 2013, just showing from about 1950 all the way up to 2007, the exponential increase in take in metric tons of grouper. And that doesn't mean that groupers are productive that entire time, right? They're being drawn down. So as fishing gear gets better, as GPS technology came about, and you're able to better predict where to go and get more and more fish, fishers, recreational and commercial, become more and more efficient at catching fish, right? And so what that's led to is that of the 163 species of grouper, 20 are at risk of extinction and 22 are considered near threatened, and many others are deemed deficient or undergoing overfishing. And so just as one example, try to illustrate what has happened to a lot of grouper is that over time, you get fewer and fewer. So the figure on the right shows, just from a spearfishing perspective in Brazil, over time, you've seen fewer and fewer days catch, right? So the population is going down. At the same time, you get fewer and fewer large individuals. So that's a trend that you would tend to see on a lot of reefs where groupers exist. And so now I want to shift over and talk specifically, after giving you a bit of background on that, on the main topic of today, which is on Atlantic Goliath Grouper, Epinephrus atadra. And so this is known to be the largest reef fish in the Western Atlantic. Its max size is 2.5 meters. I think the largest one that we've caught in our studies have been pretty close to that, 238 centimeters, so just under 2.5 meters. And so they do get that large. I'll talk about the fact that their population is still very young though since the recovery, right? These are not old individuals, they're just large. So generally the historic distribution of Goliath throughout their range. First, there was a distinction between the Atlantic Goliath Grouper and the Pacific. So that, I think it was 2008 or 9, Matthew Craig did genetic testing to differentiate the species into Epinephrus atadra and Quincunx fasciatus. So now there's Pacific Grouper that was once considered the same species. And the population was driven down from overfishing and loss of habitat, especially mangrove habitat in Florida. And so that was driven down to near extinction levels until about 1990. And since then, there's been recovery going on. And so the primary center of abundance and recovery is Florida. And that's where I'll be focusing most of my talk on today. But they range all the way down through the Caribbean and down to southern Brazil. So we have a lot of colleagues that we work with that are in Brazil that have entire groups of research where they only work on Goliath groupers. So it's a pretty big deal there. And they have regulation there, but no enforcement. That makes sense. The management plans, but no enforcement. So they don't have the same level of ability to manage as we do. And then there's also the coast of West Africa, right? And that status is fairly unknown too. My PhD advisor, Felicia Coleman, and a colleague published this paper based on anecdotal evidence that Goliath grouper may still exist on the coast of West Africa, although for a long time they were thought to be extinct there. And so this paper was based on anecdotal evidence from fishermen, from divers, from Juan himself, who spent a lot of years there in all these different countries around West Africa. So we actually have some plans to potentially go there and try to get genetic data and some other data from there to validate or verify rather that they are indeed the same species. So that will be important. And so just generally to wrap your heads around the life cycle of Goliath grouper, and some of this is very also common for most grouper. Starting with the bottom right part of this, we have I should probably step over here, might be easier. So they're from the adult spawning aggregations, right? This fish forms these large spawning aggregations offshore, and so as reproductive adults, they release eggs and sperm, right? Broadcast spawners into the water column. And then those fertilized eggs become pelagic in the open ocean for 30 to 80 days. And so that is very typical of groupers in general. And then if they're lucky enough to make it from offshore, from currents and temperatures and survival from feeding, we eventually settle into mangrove leaf litter. And in the case of Florida, a lot of that ends up being in the 10,000 Islands or in the Everglades. And so once there, they live there until about 5 to 6 years, again, if they're lucky enough to reach those older ages. And then they start to move off of those sites, right? As sub-adults, they become reproductively mature at that age. And then they move off and they eventually join adults and responding aggregations, and the whole cycle continues. So problem being, and I promised you my title that the controversy would come up periodically throughout this, and then I'll talk about the data that we've created to try to answer a lot of the gaps in data. that take increased primarily during the 1980s. And this was with the advent of a lot of spearfishing, diving technology, GPS understanding their spawning aggregations. They got targeted hard. And we worked with some of the people like Don DiMaria, who some of you know, that actually were there doing some of this and called attention to the fact and actually led the closure of this fishery, right? Because they saw that they were being fished to near extinction levels and weren't seeing them anymore. And so eventually, in a very short period of time, the whole fishery was shut down federally and state by 1990. And then controversy arose after partial recovery, right? So this happens a lot of times when you have predators on reefs, and then there's a syndrome called shifting baseline syndrome, where newer people that are in the system don't realize that what they're seeing now is not how it always was. So in this example, Goliath Grouper are a native species, right? And people consider them to be a nuisance or invasive species, but they don't realize that, 50, 60 years ago, they were much more abundant than they even are now. And so that's where a lot of the controversy comes in. And so this is a picture from like the 50s or 60s of what a typical day of spearfishing Goliath Grouper would be like. So pretty easy to get if you approach them. And so just a quick timeline, because there's been a lot of changes in their conservation status over time. So in 1990, that peak when they were brought to near extinction, there was a moratorium in US waters, right? And then following the Caribbean Fishery Councils closed in like Puerto Rico, US Virgin Islands in 1993, in Brazil in 2002. And then in 1991, NOAA Fisheries identified Goliaths as a candidate species under the Endangered Species Act. So they weren't listed yet, but they were a candidate, meaning they were being looked at as something that could eventually be completely protected. And then by 96, they were listed as critically endangered by IUCN Red List. That's the group I mentioned that I work for. And then they started to recover. So they completely were close to fishing, and they did show a trajectory of recovery. And I'll show some figures on this later. And so at around this time, then they were removed from the ESA candidacy and removed from the species of concern list. This is a good thing, right? You don't want species necessarily to be on the Endangered Species Act list unless they need to be. So they were showing recovery. And then there were some criteria differences that happened, some more stringent criteria with IUCN that then changed them into the status of vulnerable to extinction instead of critically endangered. Is still not out of the woods, but better. And that's primarily because of the recovery in Florida. They're still doing very poorly in every other place throughout the range. So they're still considered critically endangered, say in Brazil. And then that brings to the current time, which is 2022 and present. Even though a lot of the data shows that this is not a viable fishery and that they haven't fully recovered, FWC made the decision in state waters only. The federal government still has not opened this up and decided not to, but they opened up a limited harvest for juvenile Goliath Grouper in Florida state waters. And so so last year was the first year for that. And I can talk a little bit about that if you guys have questions, because that didn't seem to be a very successful opportunity for people. Only 25 of 200 tags actually got used. And so they'll be doing that again this year. So I'm very curious to see how that goes. So broadening into some of the primary research questions that over the last decade or so that colleagues and I have answered, because part of this too is that when Goliath Grouper got protected in 1990, there was literally nothing known about the species. There were a couple of old papers that had looked at some of their life history and stuff, but there was a lot that wasn't known. So it was a wide open field to answer all these questions related to their ecology and their recovery. And so some of the major questions that we've been asking and been published on since are just basic abundance and recovery patterns, age growth and reproductive physiology. There was one paper on this from before they were fished to near extinction levels up until recently, diet and trophic ecology, spawning behavior and movement patterns. So where are they spawning? Where are they coming from to get to these spawning aggregations? What are they doing during these spawning aggregations? And then health physiology and toxicology. So things like mercury is a big thing that I've studied, and how does that then affect their health and the reproduction, which it does very, very much. And then we're kind of getting into a social science aspect right now, trying to understand perspectives and perceptions of Goliath Grouper by different stakeholder groups. So that's something we're currently working on. And so this work has brought us into working on juveniles and adults. So I talked a bit about their life cycle. So you know that they're in the mangroves as juveniles and offshore as adults. So in the mangroves, and I wanted to make the point too of the importance of mangroves again, is that an estimated 75% of game fish and 90% commercial species in South Florida are dependent on mangroves for at least part of their life cycle. So that's pretty important, right? And so when we're developing and cutting down mangroves, it really needs to be a consideration in Florida that we're dependent on these mangroves for a lot of our species that we utilize. And we have over 1 million registered vessels in Florida, so a lot of people are using ocean as a resource in Florida for fishing and other things. And so this is a picture here at the bottom on the left side of life using prop root structure, which is an ideal situation. This looks a lot like the study when we go out with John and Sophie and Jen's group to look at juveniles in the Everglades. It's pretty clear, right? You can get in there, you can do surveys in the water and some other things. The two right pictures are from 10,000 Island. It's a lot murkier water. It's not a place you probably want to get in the water and do these sorts of surveys. You wouldn't see anything. And there's also a lot of juvenile bull sharks in particular in these areas. So when we were catching them using set lines, which is just putting hooks on prop roots and waiting for something to get caught, a lot of the bycatch was bull sharks like the one shown on the right. So it, again, emphasizing that it's used by a lot of different important species that are important ecologically. And I showed you the picture of the spear fisherman before, when you can target these fish and then kill a lot of them. Well, the same thing goes for research. If you know how to catch them and you know the spots to do it, like on spawning aggregations, you can catch a lot of them very rapidly. So this is how we would catch them offshore, just using hand lines, right? So just a rope with monofilament and then a hook at the end. And when the fishing was good, we would catch more than we could even process in any given time. So in this picture, you know, we're doing non-lethal sampling of everything too, right? So we have to work these fish up very quickly to sample them and then release them. And this is Bob Ellis from FWC, if anybody recognizes him from way back in the day. And so then we safely bring the fish on the boat. What I'm not showing you here is the first thing that we do, because they come up from depth. We have to vent them, right? Which means that we have to put a small hole using a trocar and cannula into their swim bladder, which heals very quickly. And that releases all the air from their swim bladder, and then they can go back to depth once we release the fish. And then we typically would strap them into a stretcher, which protects the fish as much as it protects us. Put a towel over their eyes and then run their gills with ambient seawater to keep them breathing on the boat. And then the fun part is collecting stomach contents. I don't know if anybody recognizes one of the individuals in this picture in this room, but you often have to contort yourself. And the way we would do this with juveniles is we use a lavage technique where you just stick a hose in their guts and they vomit up prey. With the adults, we found it much easier to actually stick our arms into the guts of the fish to pull out what they're eating, right? And you can actually get a lot more that way because you can feel every part of that. So thank you, Natasha, for your service. And then using aging growth, everybody's probably familiar at least with otoliths, right? It's the ear stone that you can take out, but that's a lethal mechanism or method rather to age a fish, much like coning rings on a tree, right? So on the bottom here, that's an otolith core. And you can see the opaque versus the translucent zones, and you can basically just count down that way. The non-lethal approach to this is using their fin rays to section those cross section, like you can see in the top photo. And then you can do the very same thing. There's some limitations with using fin rays, which we've come up against towards later ages, but generally it was very consistent with otoliths when we compared. And you can see here, this is after a year catching the same fish. If it was that one or another one, I don't remember, but they heal over pretty quickly. So it looks pretty brutal from the picture on the top, but they actually heal pretty quickly for this. And the alternative is much worse, right? We do a lot with their blood, and in our lab in general does a lot with blood physiology and in chemistry. So blood is a very important parameter because you can, use blood for a lot of things, like when you take your dog into the vet and look at different blood parameters, right? Calcium, cholesterol, sodium, potassium, all those sorts of things, immune system parameters. You can do that by spinning down the blood and looking at their plasma specifically. You can also look at whole blood for mercury and other toxicants, and that's generally what we did with the blood. Or stable isotopes. There's a lot of different things you can do and look at, and that's a very non-lethal way to answer a lot of questions about fish. And then all the basic tagging, sampling, and measurements, right? So length, size, weight in some cases. Bottom left picture is pretty grainy, but that was just after implanting acoustic transmitters, so tracking the movement of fish. You do a minor surgery on the fish. The top left is using a vacuum pump to pump out gonads. So instead of doing histology where you would kill a fish and take their entire gonad section, in this way, you can actually get a sample of through their gonopore and get tissue sample that way. You can't answer all the same questions you can if you have the entire gonad, but it got us to a lot of the reproductive and the physiology questions we were after. And then some basic samples for genetics and stable isotopes. And so getting into some of the results of this now, right? So one of the really cool things, and this is showing the pattern of recovery, right? I talked about some of the management plans that have gone into place since they were closed in 1990, but you can see a general recovery of the fish over time. And what's cool here is you also see the bars that are red are the spawning months, right? So that's a really good time to get a good indicator of the species and population estimate, because they're all coming together from all over the place into certain spawning aggregations. You can get a really good population estimate. But you can see the recovery that as it happens, even in the green months from non-spawning months. And then these data we found were pretty useful. They were from Reef Environmental Group, and they just collect dive data. So Goliath Grouper is a good species candidate for using data from Reef because you can't really misidentify, right? It's not like some other smaller or difficult to identify species. So we're pretty confident that people were identifying Goliath Grouper appropriately. But this just shows the general rate of recovery that happened after. And one of the really cool things that happened from this recovery is that a lot of dive operators, and this is just one example from a major dive operator off of Jupiter, where a lot of these spawning aggregations are, is the revenue shot up, number of divers shot up specifically to come and see Goliath Grouper, right? So people come from around the world now, they pay a lot of money, they come just to see Goliath Grouper. And so that has been an economic boon. And so this is one of the things that we're considering when we talk about Goliath Grouper protection and further conservation from being harvested, is that the dive community is benefiting a lot from these species, right? And the catch and release fishery. So here so on the right, you can see, kind of get an idea of what a spawning aggregation actually looks like, right? When they're healthy. And this is like, a maximum. So I'd say in general, from 10 to 100 individuals, what you would see on a reef. And this is what it would look like in a very good aggregation. And this map on the left is what I'm showing you from a paper we published, which is not showing there in 2019, was showing some of the spawning aggregations that we identified at that point. So some are still unconfirmed, and there's a lot more unconfirmed that are not on this map currently that we know about. And so you can see that they're pretty concentrated in certain areas. So in the Gulf of Mexico on the west coast, in a similar latitude, right offshore on the east coast in the Atlantic. So off of Jupiter, West Palm Beach, that area. So these are much easier to access if you're researching than those. Some of you in the room know how long it is to get a boat ride up to some of those central Gulf of Mexico sites. But really cool that we've been able to identify a lot of these spawning aggregations. And then interestingly, in this paper, we also found one, so this is a tropical, subtropical species, but we also identified a spawning aggregation far north, close off the coast of Tallahassee. And so we don't know if that was a new aggregation that had formed with climate change is one scenario, or if we had just never looked there before, which is very likely, because a lot of the areas that we looked at for unconfirmed spawning aggregations were from what fishers had told us, right? So it was kind of going there and trying to identify. So pretty cool. And so we don't know where they're coming from, where they're going. They also exist all the way to Louisiana, throughout the Gulf of Mexico, Alabama, and Texas. So they could be on oil rigs. This is also an area where there's a high level of mercury concentration of fish in the northern Gulf of Mexico, which was similar to the ones in the Atlantic. I'll talk a little bit about. And you can also see they're pretty docile most of the time, right? They will swim away from you when they get spooked, but they're just trying to chill and then like get out of the current generally. Like even in this picture, you can see they're all at the bow of this boat. And this is from the East Coast where there's really high currents, and they're literally just positioning themselves to stay out of the currents so they don't have to expend a lot of energy swimming. And so they're doing this in a berm off of Palm Beach site too. And that's just a cool picture that we took. And so this will give you a little bit of an idea of what an aggregation actually looks like without just a picture, because I think it just does it a lot more in a video. So this is one of the major spawning aggregations called MG1. This is what it looks like in two spawning aggregations. Anywhere from like 20 fish, and they're all coming all over the place. It's a degree for reproducing. And then when you get just outside, So this is the same exact site, just a month later, and you'll see what happens after they start to go back to their home sites, right? They leave the aggregations. You can see one of the fish in the background there with a notch out of its fin. You see that? There's one that we had just tagged. I'll show you a picture right there. All right. But anyway, so all these sites, even the aggregation sites, there will be residents that live there year round. But all the other individuals are coming from nearby sites or as far as 500 kilometers away. And so here I showed this picture too, if you didn't catch that. This is from one of our colleagues that's just a photographer in the area. And I just took put a couple of arrows there showing the fish just to prove to you that after we take out their fin rays, you can see the notches there, right? All those arrows. And then after we sample them, after about a day of sulking and sitting on the bottom, they'll go back and start swimming around. And we studied this, right? We wanted to make sure that our sampling techniques were harming the fish. Okay, so this is getting into some of the reproductive biology, and I'm gonna just kind of give little short segments of all the papers that we published recently to talk about this. It's all cool, but I don't have enough time to get into a lot of detail. But the main takeaways from this is that, so I mentioned that all grouper are thought to at least be able to change sex from female to male at some point during their life cycle, right? Called protogynous hermaphrodism. So what we figured out from this paper, this is led by Deb Murray at UF on age growth and reproduction, is that they're functionally gonochoristic, means that they've genetically identified sex at birth, but with the potential for diandric protogeny. So diandric just means they can either be male at birth as primary males, or they can become males later in life after they've changed sex. And what's cool here, if you look at this figure on the left, is that so males and females, so females on top, males in the middle here in turquoise, with age on the x-axis, right? Number of individuals on the y-axis, they overlap almost entirely. So there's not a lot of difference in males and females with age and how many there are. The interesting thing is looking at the transitional one. So I said about 5% of these fish are undergoing transition or have transitioned. And we know this from validated from catching them first as females and then later catching them as males, or in looking at their gonad histology, we're able to identify both male and female gonads in one sample, hermaphrodites. And what's cool is that they all overlap. There's not like one size. So even some of the very young individuals, as young as 5, are undergoing the sex change, all the way up to full adults at 20, which is about the oldest we found in the population, right? And then interestingly too, looking at their age and growth and length, is that there is a bit of sexual dimorphism going on in the population, at least on the East Coast. This is for the Atlantic population, where females are significantly larger than males as they get over 10 years of age. So, which all these things add up to further defining these as functionally gonochoristic, even though they have the potential to change sex, because there's an even 1-to-1 sex ratio across the board, and they overlap in age and size, in age and size of male and female, and small young mature males. There's quite a few of those, and low rate of sex change. And so all this means that, you know, in a population where you had more sex change going on, you would expect males to be the oldest and largest in the population, right? Because that's the only way to become a male. Clearly, that's not true because we have a lot of younger, sexually mature males. The consistent 1-to-1 sex ratio is very consistent, like very consistent. And yeah, and then just overall finding a low rate of sex change. So this is pretty cool. These questions have been asked for the last like 10 to 15 years. We finally have all the evidence put together and published it. Looking at these spawning aggregations is one of the coolest things. So all these different avenues that intersect. So looking at the histology on the and a lot of this also these patterns occur on lunar cycles. So a lot of other grouper actually will spawn and have spawning peak spawning rituals on full moon. Goliath Grouper particularly target new moon nights, the darkest nights of the year. The thought behind this being that they've evolved to behaviorally release their eggs and sperm in the water column at night, so that to reduce the amount of egg predation that happens, right? So that makes sense, right? So if you're they have I don't know if I have any picture of this, but there's a lot of these scab, these little egg predators that hang around them as like floating structures all the time, and those are known egg predators. So if you're gonna try to have maximal reproductive success, this would be a strategy. So a lot of the biology I'm gonna tell you about right now revolves around new moon dark nights. So here, this is one of the evidence. We have multiple lines of evidence for spawning aggregation. We have sound production, we have numbers increasing during peak times of year. We have the histology and then some other behaviors, but these are the things that we pinpointed. So when you look at eggs that we've collected from that vacuum pump method that I showed you, you get hydrated oocytes, which is when they put a bunch of village in and water into and hydrate their eggs just before they release them into the water column so that they float. So that's what this looks like. So that's evidence that they are just about to spawn. They're imminent spawning within 24 hours. And then just post spawning, they have these post-ovulatory follicles left over, which are the spots where the eggs were. So those are two lines of evidence that like spawning either just happened or was about to happen. So if we get that, we know that they're spawning at that time, right? We publish this in multiple papers at this point. The cool part is if you then look at the lunar cycle, and this is a map showing moon phase relative to POS and the hydrated eggs, so these two things I just described, the percent of this happening during new moon is much higher than all the other phases of the moon. So we know this is happening on new moon. And then we also put down sound recorders called hydrophones to record the sound of these fish that they make, and they will make these rapid sounds when they're spawning, right? So they make the same low frequency pulse sound at about 100 hertz year round. But during the spawning season, they do it very rapidly. And you can hear it just goes off. It's just like a chorus going on, like a cacophony of sounds. So you can see kind of what that looks like in terms of frequency here, where there's just constant pulses. You don't know necessarily how many individuals are there. That's why we get in the water and count them too and do population estimates. But it shows you that spawning is going on when you get a lot of activity. And looking at, and this is what it looks like with the pressure up top. So you see these big pulses of energy or pressure. And then this is what it looks like with the wave liking, right? So what's interesting about this too is that these are also peaking during the new moon. So at the same time when I showed you in the previous slide, where the histology shows that they're spawning at those times in peak form, they're also producing sounds at the same time at the highest frequency. So we know a lot of activity is happening on these new moons. And for those of you that have ever gone to dive with Goliath Grouper and you found one of these booms, it feels kind of like the sonic boom to your body. It actually like rocks your bones. It's so strange because they also do it for agonistic purposes, right? If they feel threatened, they'll boom at you too, or just be territorial. And if I didn't mention it already, the peak spawning times of the year are about August through mid-October. So August and September are their peak spawning months. And so when we go to study their behavior, we want to know about their reproductive spawning and what's going on in the population. We usually go out during those months to get the most information. So this is a paper that was Bob Ellison worked a lot on this paper to look at their spawning migrations and movement. So back in 2010 to 2013, 50 adult Goliath Grouper were tagged with acoustic transmitters, or these are the pingers that if you know about like the fact array, that is a grassroots movement where people from all different agencies put these acoustic receivers on the bottom of different areas of interest. So this is all in Jupiter itself on the East Coast that I'm speaking about. And all those black dots are receiver locations. So what happens when fish get pingers or acoustic tags put into their bodies in that little surgical procedure I showed you, that every time they swim past one of those receivers, then it records that fish is present in that spot. So you can tell a lot about where a fish is at any given time. And so that's how we were able to track the movement over years from 2010 to 2020. And some of the highlights of this study, the one that was just published last year, is that things that we already kind of knew, but we put together, right? They have very high site fidelity to home ranges. So they and juveniles do the same thing, right? They stick very close to their home site. They don't move very far from it at all until spawning season happens, right? So they will travel up to 500 kilometers in some cases, which I showed by that red arrow there, to reach some of these spawning aggregations down in Jupiter. And so they'll move very large distances, long distances during certain times of the year, but then they'll go back to their home sites and stay there the rest of the year. They also, this was an interesting one, is that we found very high interannual spawning site fidelity. So that means that these fish were returning back to the same spawning sites year after year after year. A very important thing. So they're not just randomly finding these sites, they're culturally finding these. They were probably shown those at some point or yeah, but it's pretty cool. And then, so they're sedentary during the non-spawning months, and then they move a lot prior to, during, and immediately after spawning. And then again, they sit tight until the next spawning season. And then the same thing that we showed here with all the other data, so this should make sense now with new moons, is that the adults arrive during new moons. That basically co-occurs with all the other evidence that we have that new moon is the most important time of year for the spawning. And males will also appear first, right? So males seem to get to the sites, and then females will follow later in a month or so. I'm not going to get too much into diet and trophic ecology. I'm just going to talk about the diet a little bit. We've got a paper that is in press right now that will hopefully be coming out in the next couple of months. But back to the sticking your hand into the Goliath Grouper. So just very basically a lot of the diet, because this becomes a big question, like what are these large animals eating? And so this is just juvenile diet, so it refers to juvenile then adult. And it's, you can see, so it's percent occurrence on the x-axis and percent mass. So the size of the prey on the y-axis and how often it occurs in their diet on the x-axis. And crabs are primarily what we're after, right? Crabs and small fish. And they're also generalists, right? So they do eat a lot of other things that they come across. So it's not that they don't eat other things. But generally crabs and small fish are what they eat. So you know, mud crabs, swimming crabs, spider crabs, all the things that you find mostly in mangrove or seagrass habitat. And then similarly with the adults, same thing. So I put a couple pictures of some of the crabs that we would take out of their guts there. And so crabs, again, are one of the highest things in their diet, right? So these are things that are outside of most of the fisheries that people complain about. They think Goliath Grouper are eating everything. Yes, they do have these negative interactions, right? I'll show you a picture in a minute. This isn't diet, of course, but fishing gear, we find tons of fishing gear in the mouths and the guts of Goliath Grouper. It occurs in at least 50% of them. And then also these scad that I mentioned that we think are egg predators occur in these tiny little things. So we don't know if they're incidentally eating them or purposely eating them, but they appear in their diet a lot. And then a lot of other things, burrfish are another big one. Some lobster. Lobster used to be a much bigger part of their diet 50 plus years ago before it became such a fishery in Florida when there's not as many of them, but they don't occur in their diet as much anymore. And then even sea turtles, and then in the paper that I'm publishing too, I talk about how like every year and every season's a bit different too. So some, like there was one year we had cutlass fish in the Atlantic from one of the upwells, and then they never appeared again. So they'll opportunistically feed on things if they're there. And so here's a couple pictures of some of the negative fishing interactions. So fishermen complained about Goliath Grouper stealing their catches, but I would argue that the result of that is far worse for the fish, right? In many instances here, some examples of some of the monofilament in a fishing line that I'll pull from their mouths and gut. I mean, in this case in particular, it was so bad that they were fish dangling in the mouth that they couldn't even get to the gut because there was such a spider web of monofilament hanging out. And it took so much time to cut all of it out. And then here was a bad example of one that I caught in one of the Gulf of Mexico that looked at it and his throat ripped. Probably it got foul hooked and then somebody powered through it and ripped its tongue hanging out. Its throat rather. So it could be pretty bad for the fish. We've also done a lot of work with mercury bioaccumulation and contaminants. So these are very large, long-lived fish, have the potential to accumulate a lot of mercury in their tissues, and indeed they do. So we looked at liver and muscle and a bunch of other tissues in these fish. And if we just look at the top right one, the important point is, so this is muscle, this is the part that you eat if you were going to eat the fish, right? Of course, they've been protected, you wouldn't eat them. But now people are potentially eating juveniles. We don't know if that'll eventually become an adult fishery too. This, I broke up subadults and adults and them to the right and juveniles to the left. So almost all of the adults are above the limits of consumption for mercury set by EPA and FDA. Of course, those agencies are always reworking their numbers and then they're going higher and higher, even though scientists say to go lower and lower. But generally, no, even some of them are up to almost 30 times what EPA had recommended in the study. And even a lot of the juveniles are above that line, right? So again, something to be aware of. And methylmercury is the most toxic form of mercury in the muscle. It's more than 90% of the mercury is methyl. This is what causes neurological issues, developmental issues in newborns. And then the liver has extraordinarily high mercury concentration, which is inorganic, but that resulted in a lot of the health issues that we were seeing in Goliath Grouper. And this is unpublished data right now, but we're also interested in how they're getting it into their systems, right? Like what of the prey that they're eating are contributing to the bioaccumulation of mercury in their bodies. And these were the common prey. And I'll again do a shout out to Natasha because she spent a lot of time grinding these fish up for stable isotopes and for mercury. If you remember that, all the hours in the lab. It's very smelly. But yeah, so we spent a lot. So these were all, so the cool and unique thing about Goliath Grouper is they swallowed their fish, their prey whole. So you can take the ones that were fresh after they just eaten it, and those were the ones that we used to then grind down for mercury and stable isotopes. And so generally, none of them were extremely high in mercury, but potentially it's because they're so large and long lived that they still accumulate a lot over time. And then to some of the health effects and reproductive effects. So this is just an abstract from one of the papers that we published a couple of years ago in 2021, basically showing that the levels of mercury were extremely high, even in blood that we looked at, muscle and liver. I already talked about that a bit. And that translated into a lot of the different physiological parameters. We looked at white blood cells and the immune system, oxidative stress, those sorts of things. And there was a ton of strong correlations between mercury concentration and health effects. And then also some histology looking at macrophage aggregates, which are commonly looked at as environmental indicators of toxicological stress. And then one of the big things that came from this study too was looking at the gametes, right? So finding extraordinarily high concentrations of mercury in eggs and sperm and the gametes, right? And this is the more vulnerable stage of any animal's life, is when they're young. I mean, that's when they're most vulnerable. And the levels that were in eggs and sperm were at the same level of pH cutoff for even being toxic to consume. So you can imagine the potential effects on reproduction that would happen from having such high levels of mercury. And another thing that was really interesting about this study is that we found that they had higher amounts of mercury dumped into their eggs and sperm early on in the season and less later. So it looked like there was sort of a dumping that went on. So potentially some of the early spawners didn't do as well as later in the months. But to get to this point, you would really have to do experimental studies or a meta-analysis where Brandon's work comes in, which we're working on, looking at all the studies that show how mercury in a laboratory setting would affect viability and offspring viability. So we're getting at that, right, Brandon? All right, so ongoing, I think I'm probably getting short on time, which I'm almost done. So this is what we're working on right now, right? So this is ongoing population estimates. So Sophie, Cami, you know the study well. So we're going on to our third year of doing mark and recapture studies in the Atlantic Gulf of Mexico, going to the spawning sites, which again are the best time to go and understand an entire population is when they all come to a couple spots, right? So you can go there and get a good idea of how the entire population is doing when you go year after year. And so what we did is we went back to a lot of the same sites that one of my PhD advisors, Chris Koenig, went to back in 2013 to do a mark and recapture study, right? So we tag fish in the water, you go back later on and you count how many fish. So typically like a Peterson mark and recapture study, if you've learned about this in ecology, you would catch an animal, a fish, mark it, put it back in the water, go back later, maybe a day later, fish, and then count how many you are tagged versus untagged. We do something as a modified version of that in water, where we tag the fish in the water, dive in, and then we go back the next day and dive with them and count how many are tagged and untagged. So it gives us a really good population estimate. And so that's what we're doing here. So there's Cami, there's me, and Cami again. So we're tagging these fish using these spaghetti tags. And we're also putting down hydrophones to listen to their sound production, and to explain why that's important. And then we're doing laser measurements as well. And Ethan, who's here, has been helping a lot with this. So we can also try to understand the size distribution of these fish over time, right? We have measurements from back in 2013. We can look through all, the fish that we caught and that we've dived and done laser measurements on. So the basic principle here is that with parallel laser beams, like in that picture on the right, we can shine those perpendicular on a fish. We know the distance between the two laser dots. We can go back in a program like image J, and then basically estimate the size of that fish based on the distance between those 2 points. Right. So between those three things, we'll have a good estimate of how the population is done over a decade or more. And this is what it looks like in the water tagging a fish. Kind of like hunting, it's not that easy. And the benefit of working in the Gulf of Mexico is that there's not a lot of current, but the visibility is really not good. So it's fun. It gets a little steep, but it's. We can show you a little bit of that one. So here's just some preliminary data from this work. So you can see here, so some of these sites were not repeat sites, right? So in blue on the left is the early Chris Koenig sites, and we published some of this work last year too, showing the difference between 2013 and 2012. Now we're looking at doing these population estimates over a longer period of time to see if that changes. All right, so we've got some new sites in the mix that are just in red. Blue is the 2013, and then green is 2022. The important point to note here is that almost every single one of these sites has significantly declined. Right, so this goes with some of the data we've shown that the population is indeed declining since about 2010. And after they've had a trajectory of recovery, something is going on where they're not doing as well anymore. It's not just an exponential increase in population numbers. So we've seen massive declines in some of these sites, like MG111 was the one that I showed you the spawning aggregation in that video. We've seen a major decline there, and these are all sites in the Atlantic. The only exception to that is one site called Gary's Graves, which is a huge natural reef, and it's really hard to get a population estimate. So this original number is only based on a diver estimate of how many fish there were. It wasn't actually a population estimate from market recapture. Some of these sites we're establishing as sites that we'll look at in the future. And then the Gulf site, there was not as much done on. We had one major site in California that we went to, and then that natural blue hole, trying to set these up as future sites that we either suspected or knew were previous spawning aggregations. This one is interesting because we only saw like 6 or 8 individuals every time we dived on it. But with part of Peterson market recapture, there might have been fish that we missed. But I am also still looking at the hydrophone data to understand, because when we looked at it or listened to it earlier, there didn't seem to be any spawning sounds going on. So I still think that that's a site that has completely been lost in the spawning aggregation. And then just to finish this off is to talk about following up with some of the controversy that's gone on. So one of the issues is there just is not, so the fishery was closed in 1990, right? I've said that a few times now. Problem is, with that comes no fisheries data, no fisheries dependent, no fisheries independent data. So we're really left with not great data to try to understand the population trends that have happened since the closure. And so federal stock assessments have been tried and have failed. Last one in 2016. The best data that we really have right now is reef environmental data, right? And we've done studies before. We've done scientific studies to compare that with reef, and they've held up, and they've correlated quite strongly. And so this figure we just published in a paper. It was supplied by Dr. Dan Greenberg on Scripps, where we took all the Atlantic data, all the Florida data. We chunked it up a few times, but basically showed that at about 2010, the population is no longer doing as well, right? And that's what this data show. So we're seeing the potential decline from 2010 and on. And then also with our market recapture data, it shows a similar trend. So it seems to be real. Adult populations remain low. We don't know what's happening necessarily with recruitment of young fish yet. Showing this figure, we've had a number of cold kills. This is actually a figure that was produced by FWI from the Southeast Data and Assessment Review, which basically tried to come up with population trends in Goliath Grouper. It's a bit messy, but this is what FWC presented at one of their last meetings. And you can see here we put the cold events that happened. So these cold snaps happened 2008, 2010 were particularly bad. And we had almost complete collapses of the juvenile fishery. And so with that, that means limited recruitment back to the adult population for at least 4 to 6 years. And so we saw that reflected in the adult population. One of the, and then I'll just say too that, we have other issues that are going on with Goliath Grouper that are not necessarily accounted for. Jen and I have been talking a lot, there's been a lot of cold water events in the last year or so, right? One in December, one in January. We don't know the impact of that. You won't know it for years to come, right? That's just the nature of it until we can sort of get that evidence out of the adult spawning population to see what recruitment looked like in the years following. But we also saw poaching, continued habitat loss, catch and release fishery. We don't know the issue, any mortality that comes from that yet that's being looked at, and then mercury bioaccumulation. We don't know the overall effects now. We know there are effects. We don't know that affects populations. One of the lines of evidence that FWC used to open up this fishery, which I don't believe is great, is that there's this uptick in juvenile populations in 2017, or I guess in 2019, and this was all based on MRIP and Everglades National Park creel data, which is the best we have for that area, but it's indirect survey data. So that means that a lot of it is dependent on fishery behavior too, and there's surveys that are done to ask people what their catch is. So generally it tracks, but what doesn't make sense here is that you have this uptick, which isn't followed by the MRIP data, right? And then you have adults, so this is juvenile, and then you have adult data, which shows a similar uptick, which wouldn't make sense, right? Because you have an uptick in the juvenile population, you wouldn't see that trend into the adult population until 4 or more years later, right? So I think the data is a bit messy and it doesn't quite make sense, but they use this uptick as the reason, one of the reasons to open up the fishery. And I think that's just a little short-sighted. And so that'll lead into some of the comments that we've made over time, and then we published this, but we also wrote, myself and my 2 PhD advisors, Chris Koenig and Felicia Coleman, we wrote a letter to the FWC commissioners making these points, right? One, that the public perception that Goliath Grouper are invasive and destroying habitat is flawed, right? We know that they actually benefit the habitat, they increase biodiversity, this has been published. And their native species important to the ecosystem. They have very high mercury concentrations that are present, that present severe consequences to Goliath Grouper and to humans who consume them. I covered that. Recruitment to adult population is limited, and viable alternative economic considerations have not been addressed. So this is specifically targeting like dive industry, right? Where there's a lot of money that comes into the state of Florida, indirect from Goliath Grouper coming back in recovery. And so one point I'd make is that agencies, including FWC, need a paradigm shift that embraces conservation to enhance biological diversity and sustainability by using the best available science coupled with a precautionary approach. So I think that's what I would leave with there, is like a more precautionary approach, more than one that's aggressive and that opens up the fishery before most scientists say we're ready. So future directions with this work is we'll continue the mark and recapture study. I see Tony Gilbert in the audience too, and he's helped a lot with seakeepers and getting us on the water for this. Thank you, Tony. Everglades National Park juvenile subadult study with FIU. So this is the study that we're potentially working on here. Social science surveys and dissecting submitted public comments to FWC. Commissioners, what's been really interesting, we've been working on this for some time, going through 6,000 comments, and so far we've seen about 6% of people that were for opening the fishery and the rest against it. So there's some offset of what the commissioners are catering to when they're making these decisions. And then also we want to see and genetically verify whether or not these Goliath Grouper are in fact in West Africa, right, and end up in the same species. So that question, I'll open this up to any questions. I put in pictures in my question slide that had nothing to do with this talk, but I love this picture with Felicia holding hagfish slime. So I'll leave it at that.